Hello and welcome to this edition of Dr. K's Psychobabble. We are going to be talking about action potential. I've already done the video on resting potential, and in this series of videos on neuroscience, one of the basic processes within the nervous system, of course, is the neuron impulse. Kind of divided into two different parts. There's the resting state, the natural state of the neuron, which is primarily a little bit negative on the inside in terms of electrical charge, a little bit positive on the outside. However, when a neuron is stimulated, either from our environment or within, or from an adjacent neuron, it begins the process of making biochemical changes on the surface of the membrane of the neuron that result in the action potential. So let's take a little bit of a look at that. We start off with the neuron here at a resting potential. We have the cell body here, this very simple drawing of a neuron. The cell body on the left, we see the where the neurotransmitters on the left hand side have come in and they've locked onto the surface of the membrane. At this point, there's still largely a slightly negative charge on the inside and a positive charge on the outside. Now I'm going to demonstrate a stimulus here. Let's say an excitatory stimulus of some kind comes along. You're getting a shot. We'll call this a pain neuron for the sake of our argument here. And that stimulus of the needle, which then stimulates the neuron, begins a process by which we have a depolarization of the surface of the cell membrane. Now that depolarization allows for ions to switch places, essentially from the outside to the inside of the neuron, and a buildup of a negative electrical charge on the outside of the cell. Now, this is the beginning process of the action potential. Once that negative charge builds up to a genetically determined amount, a certain amount of electrical charge, that groups down at the axon hillock. This is the part of the cell body that is attached to the axon, which is where the neurological signal is going to be traveling. Once it gets to a certain buildup of charge, we have the release of the action potential. As soon as that happens, the cell body repolarizes and the sodium-potassium pump returns the surface to its resting potential. However, the negative charge has now built up over here on the beginning of the axon, and through a process that we'll be talking about later called saltatory conduction, there's a sequence of events where the electrical charge depolarizes a certain section of the axon, then the sodium-potassium pump repolarizes behind it, and then the electrical charge moves on, depolarizes, sodium-potassium pump repolarizes, and it goes on traveling down the length of the axon. And this is how the signal is being translated down the axon from one part of the nerve to the other. Now at this point, this nerve can activate a muscle, it can activate the next neuron, it can trigger the release of a hormone, it can trigger a thought, an experience of pain, whatever the neurons do. This basic structure of action potential is in every single neuron in the body. It is essentially the most common way in which neurons communicate with one another. We measure this potential by having an electrode inside the cell and an electrode outside the cell, and we measure the difference between those. So we have two electrodes, one is inside the cell and one is outside the cell. So the inside cell, once you'll see in just a little bit, the inside, the electrode inside, as soon as that action potential starts, it should see a more positive charge on the inside and a more negative charge on the outside. They will react that way, and the difference between them is then graphed on paper, and we can see that right here. So when we have essentially a cell at the resting potential, right here, there's a stimulus, and that stimulus causes the inside electrode to measure more positive charge, and you'll see that going up there. Then as soon as that happens, it repolarizes and brings that negative charge back inside of the cell. And what we also see is a process that we would call hyperpolarization, and that is there's a period of time in which the cell will not be able to fire off again. It's kind of stuck. It doesn't have enough going. It can keep on getting stimulated, but the process has to give itself some time to settle down. And this is what we call the refractory period. So that refractory period means that that neuron can no longer actually respond to any stimuli. So that is essentially the action potential. And what we want to do is generalize that to all of the neurons in the body. This neuron could have been receiving information from an adjacent neuron and passing it on to yet another adjacent neuron. It could be neurons in your brain processing a thought. There's an image from your optical processing lobes, sending that to your prefrontal cortex for processing. And it's that same bioelectrical chemical process, and this action potential then return to resting potential is constantly happening. Now I went through that very slowly. This happens in microseconds. You know that these things happen. The speed of the nervous system basically enhanced by this process, and by the process we'll be talking about later, which is when we account for this propagation of the neuron impulses when the actual axon is myelinated. It's a process by which the neuron becomes more efficient at transmitting that electrical impulse. So that's it for this edition of Psychobabble. Be sure to click on subscribe, and I will see you next time.